Hello everyone, my name is Ozge Gizlenci, a final year PhD student at the Abraham Institute of University of Cambridge. In my talk, I will be presenting my PhD project, which is about the characterization of full-length transcripts with a novel tool called Nexons that we developed in our institute, and we uncovered the regulation of poison exons in splicing factors in human germinal center B cells. Alternative splicing is an important part of RNA biology. It increases both transcriptome and proteome diversity in a given cell by generating multiple isoforms from a given gene. This occurs with the help of differential splicing patterns, which influences mRNA stability and mRNA abundance in return. It affects post-transcriptional regulation and produce diverse protein isoforms, which has a profound effect on cell fate decisions and differentiation. Alternative splicing plays a major role in the differentiation of immune cells. Previous studies showed nearly 30% of alternative spliced genes were specific to immune system. Although the role of alternative splicing is studied extensively in T cells, its role in B cell maturation and activation is less characterized. So I would like to describe the B cell maturation during an infection briefly. So during an infection, antigen-activated B cells enter to germinal center reactions. Germinal centers are microstructures which are found in the secondary lymphoid tissues, and they are composed of two compartments called dark zone and light zone. After antigen activation, the B cells which enter the dark zone proliferate and undergoes the somatic hypermutation, which increases their antibody diversity. Then they transit through the light zone in order to get help for survival signals from T follicular helper cells in order to go through a process called positive selection, in which the affinity maturation happens with the help of a transcription factor called CMYC. At the end, the B cells terminally differentiate into either memory B cells or plasma B cells with high affinity antibodies. Today I will be concentrating on the changes between naive B cells and the dark zone and light zone germinal center B cells. In our experimental design, we used human tone cells from 5 donors, and we isolated both naive B cells and germinal center B cells population using their specific markers CXCR4 for dark zone and CD83 for light zone. And we sorted them in certain number of cells per population, then isolated their RNA and used the same RNA sample for nanopore long read sequencing and Illumina short read sequencing. We first checked the alternative splicing nuance, which were significantly changed between naive and dark zone and naive and light zone populations. We performed RMS Analysis for short read data, which is a tool for alternative splicing analysis for specific to short read data sets. As you can see in each pairwise comparisons between naive and activated cell population, we are able to see robust changes for each differential splicing patterns. In our dataset, we have a gene of interest, PKM, which is a pyruvate kinase encoding gene. And we know that this gene is differentially spliced, and it has 2 protein coding transcripts, which is generated with the mutually exclusive use of exon 9 and exon 10. And these 2 isoforms are important for the metabolism of germinal center B cells. Visualizing the reads and IGV show the existence of PKM alternative splice isoforms, PKM1 and PKM2, and this was confirming that we are able to capture those isoforms with our raw data. So overall, we can say this individual long reads can give estimate of the differentially spliced genes in each condition. Next, we performed an isoform detection analysis with a widely used isoform detection tool, FLAIR, to quantitate and identify the high confidence isoforms. We used default stringency of 10 supporting reads per isoforms. We were able to detect PKM2 isoform, while PKM1 isoform was not detected with default stringency, even though the raw reads showed the existence of PKM1 isoform structure. Decreasing stringency didn't help to show the correct proportion of the PKM1 existence, which was already was supported by our previous finding of short read data. Although we have found that FLAIR is a more sensitive tool than existing long-read tools, individual investigation of the genes with known isoform structures revealed inconsistencies with FLAIR-detected isoforms. Therefore, we concluded that there is a need for a pipeline which can identify alternative splicing isoform in a more accurate and comprehensive manner. Then we invested on developing a new analysis pipeline, Nexons, for the detection of transcript isoforms from long-read data. Here I would like to introduce our Nexons pipeline to identify splice variants from long reads. We call this pipeline Nexons, which stands for nanopore exons. So the pipeline starts with map long reads using a third-party tool. In our case, it was Minimap2. Then, using a quantification tool, we detected first highly expressed genes. Using an in-house developed tool, Exons, we define genomic regions of each gene and start realigning the cDNA sequences to genomic segments. So we can extract splice junction coordinates, which will be further used for defining the isoform structures. Using our Nexons tool, we aggregate similar structures after feeding them with known isoform structures. Then we filter identified transcripts by their read counts. Next, we quantitate and compare this variance between conditions by using our R-package Nexons analysis, which is another tool that we developed for further downstream analysis for differential isoform use. Using Nexons, we identified the transcript variants of our positive control gene PKM. Here you can see the isoform structures represented according to their splice junction positions in the genome, and isoforms are colored according to their stand direction. In this case, PKM is expressed on the sense strand. PKM2 together with PKM1 was detected as top 2 expressed isoform variants of PKM gene, and their proportion was correlated with our previous finding from the short read data using germinal sequencing. During the isoform detection process, one of the major challenges is truncated reads. This limits the accurate detection of isoforms from long-read data. As RNA is prone to degradation prior or during the library preparation, it is difficult to separate these truncations are due to sequencing artifacts or they are from biological truncations. Therefore, we decided to implement a flagging option to Nexons for detecting the isoform variants, which might be the truncations of other transcripts. So in this case, these two variants seems to be coming from either these annotated transcripts or from another novel variants, which might be an interesting one, but seems to be very similar structures. So it will help us to concentrate on high confidence known isoforms by filtering out these truncated isoforms. Next, we leveraged the existence of spiking control RNAs in our dataset to test the sensitivity of isoform detection by Nexons and FLAIR. SIV spiking RNAs are composed of 69 transcript isoforms which are spiked in at equimolar concentrations. They are encoded by 7 artificial SIV genes, and they mimic the human transcriptome with the complex isoform structures. which makes them really a good tool for benchmarking isoform detection tools. With that, we were able to detect 50 out of 69 SIRB isoforms using Nexons and 44 out of 69 total SIRB isoforms by using FLAIR. We then compared the quantification of the detected isoforms in both tools, and we demonstrated that Nexons can detect isoforms with higher read counts in a uniform trend, while FLAIR showed a lower read count distribution quantifying these isoforms. Then we checked if there is a bias in detecting isoforms according to their transcript length. Correlation between the quantification and the transcript length suggested that FLAIR has a slight bias in detecting the isoforms with a longer transcript length, while we couldn't see the same kind of bias in Nexons. After setting the whole workflow, we proceeded for differential isoform analysis with a comparison of germinal-centered B cell populations to naive B cells. For this talk, I will mostly show the comparisons between naive and lysosome germinal-centered B cell population. However, I would like to indicate that we observed comparable results for the comparison between naive versus dark zone. Because we are interested in MYC-regulated positive selection process in germinal center, we first checked the differentially expressed and spliced MYC target genes upon activation. Among 200 MYC hallmark genes, 44 of them were differentially expressed and 31 of them were differentially spliced. Among these three overlapping genes, we found that a serine-rich splicing factor, which is SRSF3, showed differential splicing upon germinal center reaction. Protein coding isoform of SRSF3 was upregulated in the light zone germinal center B cells, while naive B cells showed a higher expression in the isoform use of a nonsense-mediated decay isoform. When we checked the structure of this nonsense-mediated decay transcript, we found that they are regulated by a poison exon, which carries a premature terminal codon, which drives them to nonsense-mediated decay. So, literature research showed that these family members are post-transcriptionally regulated by alternative splicing coupled to nonsense-mediated decay, with the splicing in and out of this poison exon. So, for protein coding isoforms to be expressed, they splice out their poison exons, which carry this premature termination codon. Next, we further look into the isoform use of SR family members among Nexon-identified transcripts. We indeed were able to see an alternative isoform use of SR family members between the light zone and naive populations. Next, we checked the significance of these differentially spliced isoforms using DreamSeq, and indeed it was showing that SR family members actually are differentially spliced among these two B cell populations. So another example for that was SRSF1 gene, which is particularly interesting since it's another directly regulated MYC target gene. So SRSF1 showed a higher protein coding isoform expression in the light zone naive B cells, and while we saw the nonsense mediated decay transcript was more expressed in the naive B cells. When we checked the expression of these two transcripts across these three populations, we could observe the nonsense-mediated decay transcript was more upregulated in the naive B cells. When the B cells enter to germinal center reaction, they lose the expression of the NMD transcript and they start to express the protein coding transcript or SRSF1, which then give rise to expression of SRSF1 protein. Next, we checked the expression of the all ISR family members across the germinal center B cell populations and naive B cell, and we could see some of the members show the higher expression during the germinal center reaction. And luckily, we found also proteome data from the same human tonsil compartments, and we could observe most of these SR family members actually showed an increased expression during the germinal center reaction, which gives us a clue about their possible role in positive selection process. In summary, today in my talk, I was able to show that Nexon can resolve the complex isoform structures from nanopore long-read data, and we applied Nexons to study alternative splicing changes in human germinal center B cell populations. We showed SR family members are post-transcriptionally regulated by poison exons during the germinal center reaction. Overall, we expect to have a deeper insight into alternative splicing isoform regulation during positive selection process by using this tool. This work was possible with the contribution of many people, so I would like to thank my supervisor Martin Turner for his great support during my PhD, and to Louise Matheson for amazing brainstorming sessions, and to our bioinformatics group Simon Andrews and Laura Biggins for helping me to develop Nexons tool together with Louise Matheson. And finally, I would like to thank Jingyu Chen, Daniel Hudson for collaborating with us on human tonsils long-read characterization, and also to Rebecca Behrens for helping me to develop long input cDNA PCR sequencing method with ONT. And you can find further details in the posters in iRNA session and also special session of computational immunology in these indicated numbers. Thank you very much for listening.